Hello students, welcome to EPG Pathshala. I am Dr. Chirantan Rawal from Dr. APJ Abdul Kalam Government College, Silvassa, that is in UT Dadra and Nagar Haveli. Today we are going to discuss about regulation of glycogen degradation, that is glycogenolysis, under paper carbohydrate metabolism. So students, let us see what we are going to learn in this module. We will try to explore the covalent modification of glycogen phosphorylase. We will try to understand allosteric control of phosphorylase B. We try to explore the role of hormones like insulin, glucagon in the control of glycogenolysis. We will try to explore the role of calcium and how it affects the glycogenolysis. Certain tissues such as the brain, red blood cell rely on glucose for fuel. Glucose for them it's a currency. Serum glucose level must be maintained at about 5 millimolar. If we do not maintain at that particular level, then we need to face the consequences. So, to maintain the homeostasis, the concentration of the serum in the blood is important. Serum glucose is maintained by dietary sources, glycogen breakdown and synthesis from non-carbohydrate precursors via gluconeogenesis. So, ultimately to maintain the serum level, we need to focus on sources like dietary sources, which provides the glucose molecule. From breakdown of the glycogen, we will get the glucose. By inhibiting the glycogen synthesis, we may make more available glucose. At the same time, non-carbohydrate precursors will tend to the synthesis of glucose by the process called gluconeogenesis. In this way, we can maintain the level of glucose within the serum. Today, I will be briefing you about glycogenolysis and its regulation mechanism. Glycogenolysis is nothing but the breakdown of a glycogen, and how we regulate the glycogenolysis so that the minimum level of the glucose must be maintained within the body. Glycogen is a large polymer of glucose residues linked by alpha 1,4 glycosidic bonds with branches every 10 residue or so via a alpha 1,6 glycosidic bonds. Glycogen provides an important energy reserve for the body. The two main storage sites are the liver and skeletal muscles, where the glycogen is stored as the granules in the cytosol. The granules contain not only the glycogen but also the enzyme and the regulatory proteins that are required for the degradation of the glycogen as well as the synthesis. So, within these granules, your glycogen is also there. The enzymes that is required for the synthesis, the enzymes that is required for the degradation is also lying within that. Glycogen metabolism is important because it enables the blood glucose level to be maintained between the meals via glycogen stores in the liver and also provide as an energy reserve for muscular activity. So, over here our concentration is how to maintain the concentration of glucose within the body, and to maintain that we need to regulate glycogenolysis, we need to regulate glycogenesis, we need to regulate gluconeogenesis. The maintenance of a blood glucose is essential in order to supply tissue with an easily metabolizable energy source, particularly the brain, which uses only glucose except after a long starvation period. If glycogen synthesis and glycogenolysis, that is glycogen degradation, were allowed to occur simultaneously, the net effect would be hydrolysis of UTP, a so-called futile cycle. In a futile cycle, what happens? The anabolic and catabolic process occurs simultaneously, and the end of the futile cycle, we lose the energy in the form of heat and we do not get anything. So, to avoid the futile cycle, it is very necessary to maintain and regulate the various synthesis and breakdown processes. To avoid this, both the pathways need to be tightly controlled. Which pathways? Glycogenesis as well as glycogenolysis. Both needs to be controlled. This control is carried out via allosteric regulation and covalent modification of both the glycogen synthase and phosphorylase. In addition, the covalent modification is under close control of hormones. So, over here, allosteric modification, covalent modification and the hormones play an important role in the regulation of glycogen breakdown. So, the covalent modification is under close hormonal control. So, what is the concept map for the today's class? First of all, we will explore the glycogen breakdown in a brief. We will try to explore the importance of regulation, specifically the concept of futile cycle. We will explore the biochemistry of different regulatory mechanism of glycogenolysis. Glycogen is a highly branched large polymer of glucose molecule. It is linked along its main line by alpha-1,4 glycosidic linkage. The branches arise by alpha-1,6 glycosidic bond at about every 10th residue. In vertebrates, many microorganisms, surplus glucose is converted to glycogen. Many microorganisms accumulates carbon and energy reserves to manage their starvation condition provisionally present in the surroundings. The biosynthesis of glycogen is the main strategy for such metabolic storage. Glycogen is a major intracellular reserve polymer. Glycogen consists of alpha-1,4 linked glucose subunits with alpha-1,6 linked glucose at the branching points. First of all, a question arises: why regulation is important? Why there is need to control? What happens if there is no control? Suppose simultaneous synthesis and breakdown of glycogen results into the hydrolysis of UTP glucose. You can see from the diagram given in the slide, it results into the futile cycle. Due to this reason, it is very important to control this pathway. A futile cycle, also known as substrate cycle, occurs when two metabolic pathways run simultaneously in opposite direction and have no overall effect other than to dissipate energy in the form of heat. So, when a futile cycle runs, cell does not gain anything, but ultimately it lost energy in the form of heat. So, it is very important to control the futile cycle. So, that's why the regulation is important. Let's have an overview of glycogenolysis. Glycogen phosphorylase catalyzes phosphorylatic cleavage. It occurs at the non-reducing end of the glycogen chain. Debranching enzyme separates the branch and transfer it to the main chain. It also releases the residue at alpha-1,6 branch as free glucose. Phosphoglucomutase converts glucose 1-phosphate into glucose 1,6-phosphate. Glucose 6-phosphate can enter into glycolysis. In endoplasmic reticulum of liver, glucose 6-phosphate can be converted to free glucose by glucose 6-phosphatase. Free glucose release into the blood. So what is the ultimate mechanism of the regulation? It is well known fact that to avoid a futile cycle, both pathways needs to be tightly controlled. This control is carried out via allosteric regulation, covalent modification of both the glycogen synthase and phosphorylase. In addition, the covalent modification is also under the close control of hormonal control. We will understand this different mechanism under different subtitles. That is covalent modification of glycogen phosphorylase, allosteric control of phosphorylase B, hormonal control, calcium control of glycogen breakdown. Within the hormonal control, we will try to explore the stimulation of glycogen breakdown by adrenaline and glucagon, while the inactivation of glycogen breakdown by insulin. Covalent modification of glycogen phosphorylase. Okay, in this diagram, you can see that phosphorylase B, which is a dephosphorylated form, inactive, is converted to phosphorylase A by the enzyme phosphorylase kinase. Similarly, phosphorylase A, which is active and phosphorylated, is getting dephosphorylated and inactive form by phosphorylase protein phosphatase. Phosphorylase enzyme exists in two forms, that is phosphorylase A and phosphorylase B. Phosphorylase A is an active form of an enzyme. You just remember A for active. This phosphorylase A is an active form of an enzyme, while phosphorylase B is normally inactive form of an enzyme. Phosphorylase B is converted into phosphorylase A by the process of phosphorylation of serine residue. This particular step is catalyzed by phosphorylase kinase enzyme. Active phosphorylase, that is phosphorylase A, that is again a phosphorylated phosphorylase, is converted back into the inactive phosphorylase B by the removal of the phosphate group from the active phosphorylase A. It is catalyzed by a protein phosphatase 1 enzyme. Allosteric control of phosphorylase B. Most of the phosphorylase found in the resting muscle is phosphorylase B. Phosphorylase B is inactive form. Phosphorylase B can be controlled allosterically. AMP activates, that is adenosine monophosphate activates the phosphorylase B, while ATP and glucose 6-phosphate inhibits the phosphorylase B. Glycogen breakdown occurs when there is high demand of ATP in the muscles. That is, low concentration of ATP and glucose 6-phosphate and high concentration of AMP. High concentration of adenosine monophosphate stimulates phosphorylase and hence glycogen breakdown. Stimulation will be reversed in resting muscles. Phosphorylase A is not affected by ATP, AMP or a glucose 6-phosphate. It remains active under all conditions. In liver, phosphorylase B is not activated by AMP and therefore always inactive. So, glycogen degradation in liver is not responsive to the energy status of the cell. Rather, phosphorylase A is deactivated by glucose. This fits with the different role of glycogen storage in liver. As you know that the glycogen is stored in the liver as well as in the muscles. But muscular glycogen is available for the contraction of the muscles, while for the rest of the body glycogen is stored in the liver. So within the liver it is very necessary there should be a strict control over the breakdown and the synthesis of a glycogen. This particular case fits with the different role of glycogen storage in the liver, namely to maintain the blood level of the glucose. Thus, as glucose level rises, glycogen degradation by liver phosphorylase A is shut off and the degradation starts again only as the glucose level falls. So, what we discuss is if glucose level in the blood is more in amount, then there is no need to carry out glycogen degradation. But instead, in that case, the synthesis of a glycogen occurs. But if the glucose level within the blood is very less, then glycogenolysis will occur, breakdown of the glycogen will occur, and which provides the sufficient amount of the glucose within the blood. The efficient breakdown of a glycogen to provide glucose 6-phosphate for further metabolism requires 4 enzyme activities. One, to degrade glycogen. Second, to remodel glycogen so that it remains as a substrate for degradation. And the third thing, to convert the product of a glycogen breakdown into a form of suitable for further metabolism. Hormonal control, that is a stimulation of glycogen breakdown by adrenaline. So ultimately we will discuss over here mechanism of action of adrenaline. How adrenaline affects the glycogen breakdown. Glycogen metabolism is strongly controlled by hormones. When blood glucose level fall, alpha cells of pancreas secretes the glucagon. Glucagon stimulates glycogenolysis inside the liver. Glycogenolysis releases glucose into the bloodstream to improve the blood glucose level again. Flight or fight response stimulates the adrenal medulla to release adrenaline. Adrenaline promotes the glycogenolysis, which in turn fulfill the energy requirement of the cell. Adrenaline acts on both muscles and liver cells. Muscle cells do not contain the receptor of glucagon. Earl Sutherland confirmed that phosphorylase B dominate in resting muscles. But during muscular action, the hormone epinephrine activates phosphorylation of serine residue in phosphorylase B, which converts it into phosphorylase A. Phosphorylase A is an active form. Adrenaline binds to the beta-adrenergic receptor on the plasma membrane of the target cell. It causes a conformational change in the protein. It activates a G protein, which in turn activates the adenylate cyclase enzyme. Activated adenylate cyclase converts ATP2 into a cyclic AMP. Cyclic AMP acts as a second messenger. The cyclic AMP binds to the cyclic AMP dependent protein kinase. The active protein kinase A phosphorylates phosphorylase kinase. Phosphorylated phosphorylase kinase is active form of phosphorylase kinase. It phosphorylates serine residue in phosphorylase B, which converts it into phosphorylase A, that is more active form. This set of reaction is called a cascade. Cascade response amplifies the response, germinates by a very minute quantity of hormones. Very small quantity of these hormones generates an amplified response, and that response is a cascade response. On a cell surface and activates a pathway that direct to activation of protein phosphatase 1. Insulin is released in the blood by pancreas when blood glucose level is high. Insulin binds to the receptor on a plasma membrane. Binding results into the activation of the receptor. Activated receptor has a tyrosine kinase activity. It will activate insulin responsive protein kinase. Activated insulin responsive protein kinase phosphorylates protein phosphatase 1. It will activate protein phosphatase 1. Protein phosphatase 1 ensures dephosphorylation of phosphorylase kinase. Dephosphorylation makes it inactive. Dephosphorylase kinase, that is inactive, is unable to phosphorylate glycogen phosphorylase B. It results into the decrease in the glycogen breakdown. Calcium control of glycogen breakdown. High concentration of calcium ions partially activates the dephosphorylated phosphorylase kinase. During muscular contraction, sarcoplasmic reticulum releases calcium ions. A calcium ions thus stimulates the glycogen breakdown. So we learn the regulation of glycogen breakdown. Ultimately, a regulation of a glycogen breakdown is very important. Glycogen is a branched polymer of glucose that serve as an osmotically neutral means to store glucose in a cell. It will be useful in times of need. It is present in organism from bacteria, archaea to humans. So the regulation of glycogen degradation as well as synthesis is must. Glycogen degradation and glycogen synthesis are controlled both by allosteric regulation and by hormonal control. Stop. So students, let us summarize what we have learned in this module. Glycogen breakdown is important for the body because as and when we require glucose, that is available by the glycogen breakdown. But at the same time, the regulation of the glycogen is also important. If we can't regulate the glycogen metabolism, it will create a futile cycle that ends into what? a loss of energy in the form of heat. Glycogen is stored mainly by the liver and skeletal muscles as an energy reserve. The role of stored glycogen in muscle is to provide a source of energy upon prolonged muscle contraction. In contrast, glycogen stored in the liver is used to maintain the blood glucose level. So, as I told earlier, the glycogen that is available to muscles is used for the muscular contraction, while the glycogen that is stored in the liver is available to maintain the blood glucose level. In this lecture, we learn about the regulation of the glycogen breakdown, how breakdown of the glycogen takes place, in which condition liver has to go for glycogen synthesis and in which condition the liver has to go for breakdown of glycogen. If a blood glucose level is high, again, then liver need not to go for glycogen breakdown. Instead, it will move for glycogen synthesis. Same is the case as we discussed. The hormones come into the action. If a blood glucose level is high, insulin come into the action and it will reduce the blood glucose level. But the same opposite effect we get with the glucagon. We learn about the role of hormones and different enzymes in the regulation of glycogenolysis. Over the last 20 years, improvement have been achieved in understanding how glycogen is regulated at the molecular level and how it is connected with other biological processes. My dear friends, still we are not clear with many other aspects of the glycogen metabolism. What are the roles of the sRNAs in the glycogen metabolism? It is not clear, but as and when the new technologies are available, the scientists are trying to explore the complete metabolism of a glycogen at molecular level. With emerging new techniques, the next decade will undoubtedly give us a better understanding of a feature which at present are not completely understood, such as the involvements of the RNA in regulation of carbohydrate metabolism in general and of glycogen in particular. Thank you very much, my friends.